Well, thank you everybody for tuning in to this live stream or watching the recording. In attendance tonight are myself, Dr. Ross Boucek with Bonefish Tarpon Trust, Dr. Mike Parsons, Director of the Vassar Marine and Environmental Science Research Field Station at the Water School at Florida Gulf Coast University, and Dr. Alison Robertson, Associate Professor at University of South Alabama and Senior Researcher at Dolphin Island Sea Lab. Last, Kellie Ralston's here as a representative of BTT, the Vice President of Conservation and Public Policy. A special thanks to Dr. Robertson and Dr. Parsons. They are two of the world leaders in studying harmful algae that lives on the bottom, the toxins those harmful algae produce, and how those toxins affect fish, wildlife, and potentially humans. At this point, I'll turn it over to Dr. Parsons and Dr. Robertson to introduce themselves. So Dr. Parsons, please tell us a little bit about your research background, your experience, and how long you've been working in Florida Keys.
Sure. Thanks, Ross. Thanks to the Bonefish and Tarpon Trust for inviting me for this live stream. This is a very important topic, as we all know. So I'm a biological oceanographer by training. I've been studying harmful algal blooms for probably 25 plus years. I've been involved in studying these benthic harmful algal blooms for about the past 25 or 24 years, actually. And so pretty early on in my HAB career. And in particular, the thing that I study is called ciguatera poisoning. And we'll be talking about the causative organism quite a bit today. It's a dinoflagellate called Gambier discus. Dinoflagellates are single-celled microscopic algae, and probably the most well-known dinoflagellate down here in Florida would be Florida red tide, Corynea brevis. So Gambier discus is a different kind of dinoflagellate, and it lives on the bottom, usually attached to seagrasses or seaweeds, whereas red tide is up in the water column swimming around more. And so I'm mainly interested in the ecology of Gambier discus, the conditions that allow it to grow and proliferate, trying to answer some more difficult questions in terms of how it behaves with its neighbors and where it likes to live, what's a good neighborhood versus a bad neighborhood. And that leads more into things like toxin production, which starts moving more into Dr. Robertson and her field. So I think that's good enough for me right now, and then maybe we'll pass it over to Al.
Dr. Robertson, same questions, research background, experience, and how long you've been working on it.
Yeah, sure. I've been working on natural toxins and venoms and related things for about 20 years. Did my graduate work on these sorts of things, but I'm very interested, and I work on how, when, and why different microorganisms produce toxins, and then how those toxins move into other seafood species and what effects they might have within those species. And that can include invertebrates like lobsters, clams, oysters, those sorts of things, but also fishes, marine mammals, and people, right, in coastal communities. So we do a lot of work with coastal communities trying to understand and mitigate risk associated with consumption of seafood that might be impacted. But we work also a lot on understanding the physiological effects, so how this changes the health and viability of different species and their behaviors as a result of that. So that's the bulk of the work that we do in my research, but very much interested in things that happen on through time and on different spatial scales, and what the impacts of changing climatic conditions might be, changing temperatures, changing storm events, and those sorts of things, so that we can better prepare and predict ecological risk in these species or human health risk.
I think it's one thing Al and I didn't point out is we've been working together now for at least 13 years, and we've been heavily involved in working in the Florida Keys for those 13 years. I've also worked in Hawaii. We both work in the Caribbean. Al works elsewhere in the Pacific and globally. So I guess our research interests are global, and we've been going at it for quite a while now.
Yeah, and I would say working on species that are pelagic, so those that are in the water column, and we also have both worked on those that live on the bottom, and have been very focused on those because they were heavily understudied for a long time. So yeah, we do both because both matter, but yeah, we're very, very interested in those that live on the bottom, and maybe have a more chronic effect in some instances. But, yeah.
Thank you guys for your description. Kellie, would you like to give a quick introduction about yourself?
Sure. Thanks, Ross, and appreciate Mike and Alison being here tonight. I'm Kellie Ralston. I'm the Vice President for Conservation and Public Policy with BTT. Have a long career in the policy side of things, but also a science background, working in marine biology at both bachelor's and master's level at Florida State University. So it's been a pleasure to work with our scientists and kind of move these issues through the policy world so that we can adequately address them. So thank you very much.
Well, thank you guys. It's we appreciate everything you're doing down here and how much time you've spent to help us investigate this. This event, this live stream event, and future live stream events will provide us research updates on the spinning fish phenomenon and the mass mortality event of sawfish in the Keys. The purpose of these is so that everybody can hear research updates as they come from the researchers' mouths, so we can move through this event together and come back in the back end as a more resilient community, and ready for the next thing that the world wants to throw at the Keys. Our engagement in this and our initiative in this really began with strong partnerships with academics, FGCU, University of South Alabama, and FIU, other nonprofits, particularly Lower Keys Guides Association and BTT, and then of course our state agency partners, Florida Fish and Wildlife Conservation Commission and Florida Department of Environmental Protection. And collaboratively, we've been working pretty much nonstop since January to figure out what is causing these fish to spin, what is causing these impacts to sawfish, what is the fishery impacts of this event we're seeing, and importantly, what can we do about it? To the best of our knowledge, this event started in early October when guides and anglers going out at night would start seeing fish spin when they shine flashlights on them, or turn upside down and spin or swim up in circles erratically to the bottom. Through November, this continued, and in early December, it seemed to intensify. At that time, Bonefish Tarpon Trust and Lower Keys Guides Association alerted our state and federal agency partners of what was going on. And by early January, research began. The research approach explored 4 lines of inquiry. Is the source of this stress, is this a human contaminant related issue? Is this some pesticides coming off the land? Is it something coming off a building, or is it something coming out of a wastewater treatment plant as effluent, or a combination of the two? We looked at fish health metrics. Are these fish just been sick for a long time and something's happened to kick them over the edge? Do they have high parasite loads or pathogen burdens, or some combination of that? We looked at whether or not this could be something in the water column. Is it red tide, something similar to that? And last, is it something toxic coming off the bottom that could be affecting our fish? To attack these research questions, we've been now on 25 different research missions since mid-January. That's about that's 1 trip every 3 days, doing something, investigating something. That's included over 30 biologists and 12 fishing guides that have committed time to this effort. Together, we've collected 150 fish samples for analysis, 200 water samples, and over 200 substrate samples to be processed. Importantly, part of this initiative is not just the field work, it's the time in the lab, which involves an army of researchers. I know in Mike and Alison's lab and in FWC's lab, they've been working around the clock to attack this problem. FWC has committed 30 researchers to it. Alison's lab is all on board, as is Mike's, to try to find answers to this as quickly as possible. For some things we've learned so far to date through our professional reporting networks, through fishing guides and charter boat captains, we've received over 200 reports of symptomatic fish. The reports cover 35 different species, from forage fish, game fish, to sharks and rays, and basically everything else in between. So whatever this is, it does not discriminate based off species, size, migratory pathway, or behavior. Up until recently, the reports were really localized from west of Big Pine to Sugarloaf, and really localized to inshore waters. Of the 200 reports we've had, only 2 of them came from Hawks Channel, the reef tract, near shore, offshore water. So really, this is an inshore seeming issue, at least at this time. There's been a recent expansion east and west that we're continuing to monitor with our partners. In addition to learning about how this is moving in space and time, what's being affected, and how that changes from day to day, our partners, and through our collaborations, have learned a lot. Florida Department of Environmental Protection has evaluated many different suites of human contaminants that could be in the water affecting these fish. This includes pesticides, human contaminant tracers, and everything else in between. And they're continuing to do this. And so far, nothing has come back beyond background level. FWC harmful algal blooms has determined there's no red tide in the water or no similar types of harmful algal blooms. And all the fish health metrics have come back fine. They're basically healthy fish that is something is stressing them immediately, causing them to freak out. And last, the water chemistry, the water temperature, salinity, dissolved oxygen, all that is normal. Dr. Parsons' team, though, did make an unusual discovery that could be related to this, or potentially not. At this point, I'll turn it over to Dr. Parsons to tell about his ongoing research, what he's finding, and some more specifics about that work. So, Mike, Dr. Parsons, if you'd like to take over.
Thanks, Ross. Sorry, I was muted there for a second. So some of the initial sampling that was done by BTT, by the Lower Keys Guides Association, and by FWC, they noticed that there was a benthic dinoflagellate, Gambier discus, that was in the water. And they also saw it in the gut contents of at least some mullet fish. And so Ross thought that that was a potential lead. And, everything else basically was a dead end at this point. There was no real data supporting any other cause. And so Ross contacted Alison and I and asked us if we thought it could be related to Gambier discus. I got a report that FWC measured Gambier discus in the water column. It's a benthic species typically, so seeing it in the water is a little unusual. And based on our work that we've done over the past decade or so, the numbers they were reporting seemed to be high. It was much higher than we've normally seen in the water. And so that had us to thinking, well, if it's high in the water, maybe it's also going to be high down on the benthos, down on the seagrass beds, down on the seaweeds. And a lot of these sites that we sampled and where these spinning fish, where this erratic behavior was observed, are near seagrass beds. So we sent down a crew in early February working with Ross and Lower Keys Guides Association to collect some of these seagrass samples, some of these algal samples, water samples, to try to get a better handle on what these benthic dinoflagellate populations were doing. We collected water and algae and fish for Al as well, so she could start some preliminary toxin work. And what we noticed was that Gambier discus abundances were high. They were anywhere from 5 times higher to about 30 times above averages that we've seen over the past 10 years or so. So higher than we've seen it. We also looked at some of the live material, and we can grab a single cell of Gambier discus, and we call that isolating the cells, and they'll divide, they'll make clonal copies of themselves and divide. And so we have these growing in culture now. And so that'll really help us understand the physiology of Gambier discus, the toxin that it can produce, or toxins and other compounds that they can produce. So it gives us more information to work on in terms of trying to solve the issue, or at least understand more about these erratic fish behaviors. So a lot of work in the field. Our crew just got back today from the latest trip, collecting over 500 samples, I believe Adam said this round. So there's going to be a lot of lab work. Al, you got a lot of samples coming your way too. And so, you know, it's an ongoing process, and we're all working real hard to try to understand what's going on in the field, and getting these data, and then getting the samples to Al where she can do her more in-depth work.
That's alarming on several reasons. But so you said it's elevated. Is this something we can like see in the water? I work with fish that are small size, 3 inches of everything under the microscope. Like, can we see this and report it, or like how does that work?
Yeah, so we're talking about a microscopic organism, and they're almost the size of a grain of sand. And if you think about red tide, usually there won't be enough red tide in the water where you can see the water turn color, turn a brown or a red, until you get hundreds of thousands of cells per liter of water. That's a lot. You know, usually with red tide, you might see 1,000 cells per liter in terms of background. So with Gambier discus, we're only seeing, I think the maximum numbers we saw were into the thousands, below 10,000 cells, definitely, per liter of water. So that's not enough to discolor the water. That's a lot for Gambier discus, but it's not a lot in terms of our typical blooming species. So that's one reason why Gambier discus was kind of under the radar here, because it really wasn't abundant like a typical water column harmful algal bloom species would be.
Yeah, it's I think Adam or a few of you guys showed me some photos and slides. They're very, very pretty little microscopic critters. Do you have any idea of like what caused this jump in the numbers? Is there any sense of that going on?
We don't really know. Again, this is something we have not documented before, so we have not seen this. It doesn't mean it has not happened in the past, but this is the first time that we've seen it in action. Typically, when you look at, I guess, some of the historical records globally, people have noticed that you'll see an increase in ciguatera in many cases after there's some kind of environmental impact to a coral reef. The reason why I bring up ciguatera is Gambier discus produces many different toxins and precursors of toxins, some of which can accumulate in fish. And when people eat those fish, if those fish have enough of the toxins in them, people can get sick, and we call that ciguatera poisoning. So in the past, people would notice if there was a coral bleaching event, or maybe a hurricane, or in many cases, a ship that grounded on a reef, that they would notice people getting sick from eating fish from that affected area. They didn't necessarily know that it was related to Gambier discus in these historical records. But ever since then, people started thinking about environmental impacts and potentially having a relationship with this. And so if we think about possible environmental impacts that we've had down in the Keys, the one that a lot of people think about would be the really hot summer. And so could the higher than normal water temperatures that we saw down in the Keys, could that have perturbed the system in such a way that Gambier discus is now at an advantage, and maybe some other things were put at a disadvantage? Because again, Gambier discus, it has a lot of neighbors down on the seagrass beds. It's also very common in coral reef environments, living on algae in coral reef environments. And so what allowed Gambier discus to become more abundant now, we're not quite sure. Does it have anything to do with the high temperatures we saw in the summer? Trying to put together the pieces of the puzzle there and you know, almost looking for clues at the scene of the crime after the fact. So what happened there? So there's a lot of digging into some of the data, and other agencies and scientists have collected water column data, temperature measurements, and maybe there's other data out there that'll be helpful for this. So we're trying to just gather all of these clues to see if we can better understand what happened and what the potential cause could be.
Yeah, heat wave was pretty gnarly. You mentioned they produce a lot of different types of toxins, right? I guess Alison will talk more about that, but do you have any idea, based off your experience, how long this will last? And or is this going to spread, or do we have any sense of that at this point, or no?
Based on our previous work, we tend to see Gambier discus being most abundant in the fall and winter months. And then it becomes less abundant as the waters start to get warm in the summer. So I would expect its population to decrease as we get into the warm summer months. So the irony here is, did the hot summer have any cause of what we're seeing now? But next, or this coming summer, may actually suppress the Gambier discus population. So that's funny how nature works sometimes.
Yeah. Well, thanks, Dr. Parsons. I had a
Sure.
chance to go out with your field team, and your crew is very professional, hardworking, and relentless. Your lead researcher, Adam Catechus, he called us on a planned trip at 3:30 in the morning. Said he's going to be down in Cudjoe Key at 8. He hopped on a boat, dove all day in 68-degree water, went out at night with us to collect spinning fish, did another dive at night, and then was up till basically midnight post-processing samples. So really appreciate his commitment, your commitment, and your team's commitment to helping us investigate this.
Yeah, thanks a lot, Ross. We're really lucky to have such a hardworking crew. That's for sure.
Yeah. So yeah, basically we all we got right now is some weird acting fish and a lead based off your long-term work and this situation that we're in. So Alison, you're the toxin expert here, one of the world's best. You said something to us when you flew down to the Keys about some of the breakthroughs you made in discovering and mapping these different types of toxic things. So we're lucky to have you and rely on your expertise to tell us what's in store for us and their fish. Do you mind providing a little background of your work down here? What kind of stuff do you do, and what have you found so far in the samples that you've run?
Yeah, sure. And yeah, it's nice to be working with such a collaborative group of people. Like when you have an event like this, I've been involved in lots of event response kinds of things. It's really, really lovely to have so many people working together, talking, communicating, and lots of different stakeholder groups like the fishing communities, restaurateurs, and everybody like really invested in helping us understand the issue. So I'm thankful to have also been down in the Keys last weekend so that I could see this for myself firsthand. That's really important for somebody like me, where we're interested in how it affects the fish and what might be going on. But yeah, what we've been doing with this issue, so you've heard a lot of water samples have been collected, sediment samples, benthic algal samples. So these are the organisms that live on the seagrass and live on different macroalgae and sediments. So we have samples of those and a variety of fish. So we've received over 100 fish so far that we've been analyzing, and tissues for all of those fish. So that represents about, 800 samples across all of those. It includes sawfish, cowfish, a variety of grunts and jacks, mullet, the mahara and ballyhoo, and silverside and snappers and so on. So that's really helpful because it helps us start to tease apart what might be affecting such a broad range of fish. It's quite, it's quite unusual, of course, to see such a broad range of species impacted and in a fairly localized area, right? So that gives us some clues. This has been happening, and centered around areas in Coogee Bay and other spots in the Lower Keys, which is really interesting. And while we might be seeing a little bit of movement of symptomatic fish into other areas, it has lingered in these central spots for a while. So that gives us some information that whatever this issue is may have been on the bottom. So we as scientists, we have a lot of hypotheses, right, that we try and test to try and understand what's going on. There's something that might be very quick and transient or moving in and out with the tides. We might have a different thought, about things like that. But so the finding of Gambier discus on the bottom at elevated levels and slightly elevated in the water column was the lead that we have right now. Gambier discus is a genus. What this means is it's like a family, really, a group of organisms. So there's actually more than one species, and that's important. Different species have the capacity to produce different toxins, different types of toxins, different levels of toxins, and not all of them are producing toxins. So some of those that are present in the water column may not be harmful at all. But what we found through our work ongoing throughout the Keys and the Caribbean is that there are a couple of species that are known to be toxin producers. And one of those we have identified in the water samples that we have. Are they at the levels that would cause these symptoms or these behavioral problems in these fish? We're not sure. We can't get to cause and effect yet. But we have detected those toxins in the tissues of a number of the reef-associated fish that we've been looking at. So what does that mean? Does that mean that we would expect more reef fish to become symptomatic? Not necessarily, right? We do know that Gambier discus and toxin-producing Gambier discus have been present in the Keys, in the last decade that we've been looking, and we have found levels of toxins. So some of those are called ciguatoxins that Mike mentioned. We've found those in fish before. So we could be looking at background levels. We could be looking at the fact that those background levels being present in fish might make them more susceptible to to whatever issue is causing the spinning. What we've also learned is that these effects can go away. So some of the work Ross has been doing and with the Lower Keys Guides Association and anglers, it appears that the fish can recover sometimes, right? So they can get into clean water and the effects can go away. So that's somewhat transient, which gives us other information that we can think about, that these toxins, if they are present, whatever the factor is causing the spinning, is likely in the water column in some way and crossing the gills of those fish, potentially causing them to lose equilibrium and have this erratic swimming and swimming upside down, breaching from the water, those kinds of things. These sorts of symptoms are common with a variety of neurotoxins that are produced by algae. So we have been looking in my lab at all of the known toxins that can cause these sorts of behavioral effects, so all of the neurotoxins. To do that work, my team, who have been working 7 days a week, 12 to 14 hours a day sometimes, involves some bioassays. So this is where we grow neuronal cells so that we can look at the effect of the water samples and everything else on those. So we extract any possible chemical from the water, from the algal samples, from the fish tissues, And we then expose these neurons to those toxins for a period of time to see how they might respond and to see if there are any, if there's any toxicity there, if we see any kind of cellular disruption or cell death, any of that. We also do a lot of chemical analysis here. So we have the expertise to do quite a lot of advanced analytical work. So we're using some tools like mass spectrometers, which we can look very carefully at the compounds that might be present in the water column. So these toxins that we know the structure of. So we've been targeting those we know of. And then with the bioassays, we compare that with the chemical analysis to identify anything novel, anything different, anything new that we may not know what it is, but we can track it based on its bioactivity to try and understand what it might be. This is how we've discovered, for instance, the toxins that are produced by Gambier discus. And that was actually a global effort that included researchers from Canada and Norway and the United Kingdom and Australia and other places. But we all came together to try and determine the structure and the chemistry and the toxicity of those compounds. So we have recently, in the last 12 to 18 months, been able to do that. And so we know what this Greater Caribbean toxin is that's associated with Gambier discus. So we've been able to do very sensitive targeted analyses in this case to look for those toxins. And we have found some of those in the algal samples that were collected from the bottom. And we've found toxins associated with Ciguatera in the fish tissues, as we mentioned before. These organisms are also capable of producing a variety of other compounds. One of those toxin suites are called mycotoxins. These are very large molecules. They're also neurotoxins. The ciguatoxins that we just spoke of, they affect sodium channels, which are really important for nerve conduction. Another channel that's really important for our ability to move and walk and do all the things we do are calcium channels. And so the mycotoxins affect those calcium channels. In both cases, they activate those channels. So they open them and allow a lot of sodium in one case or calcium in the other to flood into cells. That causes problems with our ability for our nerve conduction to happen in a normal way. Anyway, those things happen. What we know is also that Gambier discus doesn't live alone. It lives in a community, just like we do, in our suburbs and whatever. But there are other benthic algal species that could be present living with Gambier discus that is also capable of producing bioactive substances that may affect fish behavior. Some of those include a group called Prorocentrum, and they can produce a compound we know as oxidaic acid. That's actually associated typically with diuretic shellfish poisoning. So we see it a lot in the Northeast and the Northwest and other places, and it's managed fairly well. But it has also been associated with bioaccumulation in some species. And we've detected that in some of the smaller fish, like the silver sides and others in this area recently, and in the algal samples as well. So that's sort of the data we have, and what does that mean? And could these toxins be causing these effects? The toxins that I've just talked about, like the ciguatoxins, for instance, we know from the work in my lab that they do cause behavioral effects. So we've done experiments where we have a diet laced with these ciguatoxins, so through sublethal or very low level, longer term sublethal dietary exposure, we see that fish become very hyperactive because these toxins activate those channels that I talked about, these voltage-gated channels. So they start having erratic swimming behavior and they do lose equilibrium. And they become less responsive, and through time they can become very lethargic and docile. Those effects are not usually transient. when we do dietary exposure. So there's a lack of data that we're trying to fill right now to see if waterborne exposure could result in these more transient effects that we're seeing in the fish. That's a little bit long-winded, but hopefully that helps kind of capture a little bit of the complexity of the issue. So we can't really talk about cause and effect just yet, but we're doing everything we can to look at all these known toxins I've just talked about. But also we've identified some novel compounds, we think, and we're trying to track those down and work out what they are and if they could be involved in the problem. So,
Al, could you briefly talk about toxins that we did not detect?
yeah, sure. Yeah. We looked for other toxins that are associated with other algal species. So we looked for the saxitoxins. These are a group of paralytic shellfish toxins. They actually block sodium channels. So my PhD work, actually, my graduate work was on that group of toxins in the tropics in Australia. But we're not able to detect any of those toxins with our methods, which are very sensitive to those toxins. We also looked for a variety of other shellfish toxins. I'm not going to list off the 50 others we looked at, but a number of those also were not detected and not present. We looked at for brevitoxins, so these are the toxins associated with Karenia brevis. And while we found some low levels, background kind of levels in a couple of fish, we didn't find any of those toxins in the water column by either strategy of testing. And so those would be the most important ones probably to talk about in terms of natural toxins. Yeah, good question.
Well, thanks, Alison, for us humble fish people. I think what I heard at least was that multiple toxins present. We have the cigatoxin that goes through the food web. That's the one that's not awesome. Can result in impact behavioral effects, but it takes some time, and it takes some time for them to get rid of it. The mitotoxin, it's something that's in the water. It can be transmittable for the gills, which may set up more of these instantaneous effects that we're seeing. We still don't know which one, if any of these are related or doing anything, and more results are needed for that. Is that a fair assumption? Is that a fair shakedown of complicated stuff?
Yeah, that's a good synthesis of that. The only other thing I think I left out, there are a couple of other toxins or compounds that are produced by Gambier discus that we've identified. Those are called gambierones, the gambierone and 44-methylgambierone. We know that Gambier discus produces these. What they do in fish is actually unknown, but we typically, when we find those compounds, mostly what it tells us that Gambier discus is present, because Gambier discus produces quite a lot of those. But I think we need a little bit more science on that, right? We need to do a few experiments to see if that could cause the effects we're seeing. There's a bit of a lack of data on that. We do know that at up to fairly high levels, those toxins have no effect on our neuronal assays that we run, but that doesn't mean that they couldn't affect something else.
So a couple unknown curveballs.
Yeah.
So some pretty ominous situations maybe on the table. So as somebody who's worked for the U.S. Food and Drug Administration, this is what they do for a living, you're the only one I can think of that would be qualified to answer this next question. Is it safe? Can we eat fish in the Keys, or what do we do there?
I think it's always a fairly I don't think you need to be a scientist actually to think about if fish are behaving strangely, that it's probably a good idea to avoid eating them. right? So that would be my advice. So although we don't know if those fish are safe to eat or not, if you have a fish that's symptomatic, it's probably best not to eat that fish at this time, right? So that would be the advice I can give, because I don't think we can say if they're safe or not. In the fish that we've detected stigma toxins in some of their tissues, they, and not necessarily safe to eat, but those are normal things for the keys. And I know that those are normal things fishermen know about. There are also things restaurateurs that, we've spoken to are aware of, and avoiding species that they know to be associated with bioaccumulation of those toxins. So yeah, I think, people just need to use their best judgment. And if fish are behaving very, very strangely and spinning, probably don't choose that fish, maybe move on to a different spot.
So yeah, so best advice you could give right now is, first of always consult with the Florida Department of Health
Yeah.
on fish consumption advisories. But second, to clarify, best thing we should do is use common sense. And if it's a deer foaming at the mouth with bloodshot eyes, don't shoot it and eat it. Same as if it's a snook spinning in a canal, don't scoop it up and throw it on the frying pan. Is that?
Absolutely. Yeah. I think both of those things are really accurate representations. Like none of us would go out and choose to eat, any other food animal if it was behaving really weird. Think mad cow, like that, if a cow was spinning, I probably wouldn't want that steak. So yeah, but that doesn't mean that the fish can't necessarily recover from this, right? And
Yeah, I guess eating any wild animal, there's always a risk, small risk of something happening.
Yeah, definitely. I think we all take that risk when we decide to go fishing and we decide to do anything in nature, right? But yeah, I think that common sense, using your best judgment, but ultimately it's up to any individual to make their choice. But yes, definitely look for those advisories if they're available. And honestly, one of the best resources are local fishermen and local fishing guides and so on, because they have a wealth of information, knowledge on this issue and every other issue associated with fish. So I work alongside fishermen in all the areas and regions of the world that we work, and it's been critical to everything we do. So yeah, it's really important.
Yeah, I appreciate you answering some hard questions there, and I appreciate your commitment to this. When you finally had when you had not finally had results, when you had results, you said, I would like to talk to people in the Keys one-on-one. And I said, great, let's set up a call. And you're like, no, I'm flying down to the Keys to talk to people. And then over the weekend, you and Dr. Parsons came down. We spent Saturday meeting with a bunch of people that have a big stake in this. On Sunday morning, you flew out, and on Sunday afternoon, you texted us a photo of you back in the lab grinding away at samples. So we really appreciate your commitment to this and making the time to come down and really being, in person, which is always the best. So thank you so much.
No, of course. And that picture was of my graduate student actually slaving away in the lab. So I was, thinking I was going in there to set up a couple of things, but they were on it already. Like, they're also really committed, like both the students, the staff, everybody. We care a lot. So we're not just up here doing this because it's interesting. We really care about the results and communicating that to everybody. So we're always going to want to talk to people directly, and so they can ask us all of the tough questions too. I think it's really critical.
Thank you so much. Before we transition into what Kellie's doing in Tallahassee to ensure that we have a resilient Keys, I'd like to say there is some optimism. As Alison mentioned, we're doing some studies to determine if fish can recover from this, what species are most likely to be susceptible or vulnerable, and how long does recovery take. So from our work, working in partnership with Lower Keys Guides Association, we have seen, yes, these fish can recover, which is not only great news in informing which potential toxin this could be, but also the status of our fisheries. If you see a spinning fish, doesn't necessarily mean it's dead, or it's a dead fish walking. With that said, we know, as we mentioned, that we can trigger these fish to elicit these behaviors with stressors. At night, shining a flashlight on a fish that was seemingly healthy before the light hit it can cause it to do these erratic behaviors. On the short term, a shark's going to love seeing that and want to eat it. On the long term, we don't know what the physiological consequences of this are, long-term health effects to reproduction. And that's not the only stress. It could be, catch and release fishing can potentially do this as well. So as we move through this event, and if we're in a place, particularly at night, where we think fish will be symptomatic if they're stressed, please avoid shining a light on them. They're trying to get through the night, and they're trying to do their best to be okay. And same as for us fishing. If you're catching fish and they're spinning on release, maybe try moving a little further down to a new place that they're not doing that. And some of the work that we've done has shown that this can be very patchy, where one spot will have it, and 3/4 of a mile, the fish will be fine. And usually the fishing was, at least for us doing these trials, was always better when there was not symptomatic fish. So just thinking about what we can do in the short term, those are two options that we have in our control to improve on this. Kellie, we'll have a few more concluding remarks, but Kellie, do you want to provide us an update on what's going on in Tallahassee and what is at stake for us in the Keys here?
Yeah, thank you, Ross. So to kind of build on the optimism campaign that you started, I think one of the themes that has come out of this conversation tonight that we've seen on the ground really is this idea of collaboration and support, not only between scientists, but also between our agency partners and state partners. And so it's been really encouraging to not only have those conversations with policymakers, but see such a rabid response, if you will, to make sure that all the resources that are needed to kind of fully assess this issue and address it are being provided. And it's been wonderful to have our state agency partners, FWC and DEP, involved, but also the legislature in particular. Given kind of the timing of this issue, it really kind of came to a head on the back end of our state legislative session, and our state legislators really rose to the occasion. BTT basically made a request and a plea and said, we really need additional resources to be able to address this. And we were greeted not only with strong support from our Keys legislators, but also from legislative leadership to provide $2 million, basically at the very, very end of the budget process during session to address this. And so we're very grateful and appreciative for that. As you have heard, we have a lot of kind of outstanding questions to be answered, not only exactly what is this, but how far has it spread? Will it continue to spread? What species are affected? And so there are a lot of questions that need to be answered. And, money makes the world go round, and it really does help having those resources to be able to address this. So we're very grateful for our support from both the Florida legislature, Senate, and House, and the leadership of Governor DeSantis and our state agency heads to kind of work through this as partners, all of us working together. It's a wonderful coalition, and we're very appreciative of that. I guess beyond that, kind of looking to the future, that funding was provided in the state budget. And so we'll have probably additional questions and additional needs as we move forward. And it's just been really for lack of a better word, heartwarming, to know that we have that support and that communication between all of the entities that are working, because it really is kind of a big herd of cats, and to make sure that everybody's talking to each other and providing information and support as we move forward. So we've been very, very, very grateful and very appreciative for the response that we've received so far. And also on the academic side, tremendous kudos and appreciation to Dr. Parsons and Dr. Robertson for them jumping into this so wholeheartedly to kind of help us all get to the bottom of this. So.
Yeah, thank you, Kellie, for what you're doing. Yeah, as a community, we'll get through it. Just got to make sure, this as it persists, we're all doing what we can. And I know you've been working really hard to make sure people up in Tallahassee know what's going on and are going to be paying attention to the keys to make sure that that we're not forgotten about as this continues.
Yeah, and to add on to that too, our federal partners have been alerted as well. Both Biscayne and Everglades National Park and the Florida Keys National Marine Sanctuary have been very responsive and wanting updates and wanting to know how they can assist as well. So it is kind of going from the science part, but also to the broader policy coalition, understanding that we all play a really important role in this, and that everybody's voice matters. So we've been very grateful.
Yeah, we should also add that we're NOAA responded very quickly too to provide some event response funding, through the harmful algal program. So that helped us get kick-started at least, right? So that we could start moving with you all a little bit. So
yeah, agreed. And thank you for highlighting that, Alison. It really has been encouraging to kind of see everybody jumping in with their boots up and ready to kind of hit the ground running and work together. So
absolutely. So quickly, we got 7 minutes before we wrap this up. Dr. Parsons, what's next for research? What are we doing? What are we going to learn? Any ideas of continuing work or next steps?
Yeah, so I think our efforts are going to concentrate on 2 or 3 different avenues. Number one, we're trying to sample outside of the affected areas, try to get some boundaries on it so we can see any potential expansion or contraction, try to understand the scope of the problem or the affected area a little bit better. As we get some of these isolates growing in culture, we can do some culture-based experiments, work with Al on that as she focuses in on chemicals or compounds of interest. We'll be providing her with material so she can link it directly back to Gambier discus. We'll also be looking to see what Gambier discus species are present. We've identified one species, Gambier discus carabeus. So far, FWC scientists confirmed that, and we're following up with some molecular work on field samples to learn more about the presence of other species potentially, and looking at reconstructing some of these past conditions, whatever historical or near-term data are available, but also looking at different samples where we can do stable isotope work, look in the sediments to see if there's any biomarkers or chemical markers of perturbations. And so just trying to see what mark was left on the environment as we had that hot summer moving into the fall and winter time.
I love it. That's cool. That's a really cool idea. Yeah, thanks for thinking of that. Dr. Robertson, what's next for you in terms of moving forward with this?
Yeah, so some of the things we're planning to do is to try and get to that cause and effect piece as much as we can. So with Mike's group at FGCU and others, like we're going to collect a lot of water from affected places, right? And we're going to try and do controlled experiments with fish from non-affected areas to see if we can reproduce those behavioral effects. Likewise, taking fish from non-affected areas and trying to see what happens, are they also affected, or does it take some level of pre-exposure to cause these effects? Or also, as Mike said, as his team isolates and grows these algae in culture, so they're able to grow them at a higher density, we're going to be doing a lot of work in the lab to determine if those are in fact producing any toxins, and if they are, we would then conduct some experiments to put those cells in controlled scenarios where we can expose them to fish as well and see if there are effects. This helps us trying to understand what happens. But beyond that, we're interested in looking at what's happening in time and space. So sampling broader areas with a level of frequency through time, so we can see, is this in fact spreading, or are we just getting increased reporting because people are more aware of it? Something that the fishing guides also brought up, right? That's important to tease apart, but sometimes difficult to tease apart. So we'll be looking, and we're not just looking for natural toxins, so I want to point that out too. So we have the capacity here through investments from our state that have allowed us to develop a pretty nice analytical facility. So as we look at these samples, we're trying to be open-minded about what the cause might be. So we are fractionating, so separating based on the chemistry of a given sample, so that we can look and with our bioassays kind of guide what might be affecting the fish. And we're moving to do that with small fish species here as well. So it's not just in neurons, so we can actually see if we can replicate that activity. So those are it's quite a lot, but those are our shorter term goals and ideas so that we can help solve this problem. One thing I know, Mike and I were we're very focused individuals, so we don't give up. We're not going to give up, and we're not, we're very much want to support and help in any way we can. So that can include, if guides and fisher anglers, are out on the water and you see discoloration in the water or any other weird things going on, like please collect some samples, like of water at least, or some of the fish that might be affected. And I think through the Lower Keys Guides Association and BTT, there's a way to get those samples to us, and we would be happy to look at them. I know FWC is also happy to look at some of those samples. So, yeah.
Well, thank you so much, Alison. And you bring up two important points. So if you see these discolorations in the water, if you see these fish acting weird, please report them to either the Lower Keys Guides Association's forum they have, our local system, or importantly, report them to the FWC Fish Kill Hotline. These reports need to get to scientists. If they're just reported on Facebook, we don't see them. So make sure, if you have my number, which I'm sure a lot of you do, text me, report to LKGA, report to FWC, however you think is the most appropriate way to get that information to us. Because like everybody said, this is our best way of tracking the spread of this and learning quickly and efficiently to move forward to the next steps. And if you see a dead sawfish, certainly contact the sawfish hotline, and the number of that is 1-800 or 472-9374. Yeah, it's tragic to have these individuals die, and if we can at least get their carcasses, we can hopefully make some meaning to their passing. And last but not least, in the short term, please give to BTT. We're running on shoestrings. Alison's tapping into reserve budgets, as so is Mike. So for this work to continue, funding is going to be necessary. So please become a member. If you want to do more, please do. Until the money Kellie talked about in July 1st, we can't lose time in disturbances or events. We need to move as fast as we can. And that means on the water tomorrow, next week, to get samples. So if we have the resources to do it, the faster we learn stuff. With that, I really appreciate everybody who's tuned in's time. And to summarize a little bit of Alison's results and Mike's results, what we have is symptomatic fish. We have elevated levels of Gambier discus in the same place at the same time. We're finding toxins in the Gambier discus and in the fish. We know that these fish can recover. This effect seems to be transient, which may tell us it's waterborne. We don't know that for a fact. But those are the high level points I brought home. Alison and Mike, is there any other high level messages to leave on, or Kellie?
No, I think that's a good synopsis that I can think of. Yeah.
Yeah, and I think it's important to note that, you know, we haven't solved this. We're just following the strongest lead we have right now. Collectively, everyone that's working on this has eliminated some possibilities, or at least their cold dead ends or cold leads right now in terms of red tide or low oxygen or low temperature, low salinity. So this just seems to be the strongest one. So that's where we're putting a lot of effort. And, you know, we're trying, again, as Al said, keeping an open mind here. Something else might show up, but this seems to be the most promising lead we have right now.
Yeah, unbiased, keeping an open mind. We're still saying DEP still looking for contaminants. We're still looking for fish health problems. All that's still going on, but this is the best lead that we have. Hopefully next time we tune in, we have some updates with some good news, I guess. And as we move forward with this. So I appreciate everybody's time, and look forward to seeing you around. All right. Thanks, everybody.
Thanks. Bye.